First, I'm going to talk about why we need sequence alignment, why biologists need sequence alignment, define the problem, and then we are going to talk about a solution and also whether the meaning of optimal solution, what does an optimal alignment mean? So therefore we are going to talk about scoring matrices, and given a scoring function, we are going to talk about an algorithm named based on dynamic programming, which can find the best alignment in polynomial time. So sequence alignment, it's very early. I mean, the sequence alignment problem has been in the area of biology maybe for more than 30 years. Since we have sequences, the problem of sequence alignment exists. But since 30 years ago, maybe there were not many sequences that we have today. Therefore, efficiency was not the main concern back then. People were doing alignments by hand. But after the databases grew, we had many sequences, and doing manually, these alignment manually is impossible. Therefore, what we do is we need algorithms and also even a polynomial running time and ON square running time even may not be sufficient to do database searches. Therefore, heuristic algorithms like BLAST and FASTA emerged. Even polynomial time algorithm which guarantees optimality, like Needleman, Wunsch, and Smith-Waterman algorithms, is not capable of doing database searches interactively. So BLAST does this very fast. So if you have millions of sequences, billions of letters in your database, doing pairwise alignment or n-square running time, a quadratic running time for doing searches is very costly. So first, let's start with the sequence alignment. Why do we need sequence alignment? One of the first information that biologists have about a gene or a protein is the sequence of that gene or protein. So when in the lab, the first thing that is discovered when they discover a gene, okay, this is the start of a gene, this is the end of a gene, the gene is discovered on the genome. So at that point, the only thing we know about the gene is its sequence. Okay, so it's when a gene is discovered, so all we know about this gene is the area of characters, which may be very long. I mean, it may be 1,000 base pairs long. So usually these letters are also called base pairs. Okay, they are called base pairs because in the DNA, we have the double helix. There's a complementary. So A is complemented with G, G is complemented with C. So there's a complementary strand to this DNA strand, DNA sequence, which complements it, so it forms a double helix. Therefore, when we talk about the length of the DNA sequence, we usually use the term base pairs. How many base pairs are there? For example, here in this short sequence, I have 6 base pairs. 1, 2, 3, 4, 5, 6. 6 base pairs. So these BP, or sometimes they use the acronym BP, it means base pairs. It's just the length, how many characters, I mean, just naming them as letters is just so computer scientists. I mean, they're not letters, they're physical DNA components, so they're base pairs, but for a computer science, it's just a sequence of how many letters do you have or how many characters do you have in this string. If you formalize the problem, it becomes like that. But semantically, they are base pairs. Now, the first information we have about the gene is this sequence. Now, but after we have the sequence, we wonder, okay, what does this gene do? Is it similar to another gene that is known, previously known, with a known function? So Therefore, we search for homology. So this homology is a new term to you. I didn't, for computer scientists, I haven't mentioned it last time when we were doing the introduction to biology part. So homology means that there is a similar sequence, or there's a gene which does the same, which has the same function in another species, or maybe in the same species that was discovered before. So there's another gene other than that which has the same function. This is called a homologue gene. Okay, homology, there is no level to homology. It's you're homologue or not. Okay, there's if there's a similar, if they have the same function, okay, they have the same function or not. Sometimes this homology term is used incorrectly by saying, for example, 80% homologue. There is no such thing. Okay, it's a wrong usage of 80%. Sometimes homology is used in synonymous with sequence similarity. So, but it's not correct. Homology is a Boolean term. And it says that are they derived from the same ancestor in the evolutionary history. In the evolutionary history, there may be in hypothetical ancestor genes, and these two genes, which are homologues, may have been diverged from these hypothetical ancestor genes. So that this is how they are related. Okay, for example, imagine the hypothetical organism, and there may be in the evolutionary history, there may be a speciation event which caused these two organisms to become two different species. However, they had the same gene, and these genes, by mutations or other sources of variation, these genes may have slightly different sequences from each other, but still in the diverse organisms, they still perform the same thing. An example for this is hemoglobin. Okay, for animals which have blood, for example, they have this protein hemoglobin, but the human hemoglobin gene is different than mouse hemoglobin gene. There are slight differences, but if you look at their sequences, mostly they are going to be the same. I mean, we have the mouse DNA, the mouse genome has the same information to encode that specific protein, okay, hemoglobin, and humans also have the same gene to synthesize that protein, but they are related. Okay, they perform the same function. They are related. So this relation is called homology. And this homology is a general term. There are several different sub-terms related to homology. One of them is, and the most used of these terms is orthology. Okay. So let me write them down. So homologue, homology, and then the subterms are orthology, orthologue genes, or orthologous genes, homologue genes. So orthologue genes means that these two genes, which are homologues, are in two different species. Like this example I gave just now, the mouse and human hemoglobin, they are in two different species. One of them is in mouse, the other is in human. So they are called, if they're in two different species, they are called orthologue genes. And there's also paralogues. And paralogue genes, it's like you have the same gene in the same organism with the duplication event. The duplication event causes a similar gene, but there might be some kind of a divergence. After duplication, some of the sequence parts may have been changed, so we have a different sequence, but they basically perform some tasks together. Like, for example, hemoglobin alpha chain and hemoglobin beta chain, they may be diverse. They may be in the evolutionary history. They may come from an ancestor, hypothetical ancestor, a hemoglobin gene, which because of the duplication event within the same organism, that same gene is copied on the genome to another location, and there are certain mutations and different evolutionary events like mutations, other changes, insertion, deletions, they occur independently on these two duplicates. So they are not, it's not like when you copy a string, if they're in different memory locations, on the PC, you can modify them separately, right? If you make a modification on one string, it's not going to affect the other string. It's the same idea here. After the duplication event, these two genes become separate genes on the same organism, and these mutation effects, they affect them separately, so that their sequences may be different. And if they're on the same species, they're called paralogs. I have a slide for this, actually. Let me just show that first. It's here. And there's an article, I'm going to post that on the website on this terminology. There's an article which describes what's homologue, paralogue, orthologue, and there's this term xenologue. It's a very rarely used that I don't even recall this definition right now. Okay, but I'm going to post this article on the course website so you can read it if you want to learn more about these terms. And here is an example that describes, so this is about a globin protein. And this is the evolutionary history tree, okay? So it's backwards, meaning that the root is here at the bottom. And it shows that there's, at this point here, we had a gene in some ancestor organism, okay? At this point, we had this gene, the globin gene. And here, at each branch, we have an evolutionary event. This event can be a speciation event. Speciation event causes these two branches to become two different species. This gene is copied on this organism, it becomes a different species, this branch, and the other branch, it becomes another species. So this is a speciation event. And sometimes we have duplication events, which causes the gene to be duplicated in that species. Okay, the gene in one species, for example, in this lamprey species, okay, in lampreys, the globin gene occurs there's a single globin gene, for example. Okay, so but at this point, in all these other species like snakes or mammals or frogs, we have two copies of the same gene. So it's the alpha globin and beta globin. And they become separate genes, they're different, their sequences diverse. So how does this happen? It happens at this gene duplication event. At this point, the gene is duplicated. In an ancestor organism, we have the alpha globin and beta globin appears. And this subtree is the alpha globin tree. This red subtree is the beta globin tree. And at this point, we have another speciation event and another speciation event, which causes the speciation between the difference between divergence frog, mammalian, and snakes. So we have at these leaves, we have this frog alpha globin and mammal alpha globin are orthologues. Okay, they are orthologues. They are both alpha globins, but they're in different species. And alpha globin and beta globin, here mammal alpha globin and mammal beta globin, they are paralogs. They are within the same, they are in mammals, okay, for example, they're in the same species, and but we have two copies of the same gene. Now, the question of sequence alignment is, let's get back to our question, why do we want to do sequence alignment? Is if you discover a new gene. I mean, when we discover a new gene, it's not like we discovered it in Mars, okay? We discovered it in one of the species on Earth. So, and the living things on Earth are related to each other. We have a lot of similarities, okay? A lot of them breathe air. A lot of mammals have bloods, okay? I mean, we have a lot of similarities. So the first, when we discover a new gene, the first thing we ask is this newly discovered gene homologous to a previously known gene? Okay, this is the question a biologist wants to know. Okay, this gene I have just discovered, is it a completely new gene that is specific to this organism, or does it have other similar relatives on other organisms? So to be able to answer this question, the tool we have in our hand is sequence alignment, sequence similarity. So what we do is, imagine you have a database of millions of sequences, or maybe 100 millions of genes. Okay, maybe not that much, but millions of, imagine we have a database, millions of genes, and a solution to this problem would be just compare this newly discovered gene to each of these 1 million, 2 million genes and determine which ones are the most similar to this newly discovered gene. So this is the tool we have, and this is the one of the most widely used still. I mean, it's an old problem, like 30 years ago they had the same sequence alignment problem, but still this tool is very much needed, okay? I mean, 30 years has passed, it doesn't mean that we know all the sequences. Still, each day new sequences are discovered in plants, in new organisms, in viruses. When biologists discover these genes, the first thing they do is, okay, is this homolog to a previously known gene? And what they do is they use BLAST for that. BLAST is the most popular tool for this purpose, because the BLAST has a database which contains a lot of known genes with known functions. So it has, you can accompanied with this huge database, you can just, it's basically the Google of biologists. Okay, it's that important. Okay, I mean, if we didn't have Google, you could not conduct any research. Okay, you are searching for a new subject, you are trying to learn new thing, you use Google, okay, or any other search engine. But Google is the most popular. But if we didn't have this search engine, we couldn't do anything. So BLAST is the same thing for biologists. So you should, BLAST is that popular, okay? Even I think BLAST, I tell this every year to, in this class, but now I'm trying to, now I'm questioning whether it's a myth or not, but I know that BLAST made it to the English dictionary as a verb, okay? Blasting a sequence. So it's the name of a tool, basic local alignment search tool, but biologists are using this tool as a verb, as an English verb, and it made it into the dictionary. It's that popular. So when you get a sequence, blasting a sequence means that using the tool BLAST to do a database search for that sequence. But do not forget the main question. The main question we are trying to answer is this new sequence homolog to an existing sequence? And if it is homolog, we also want to find out which regions are conserved. So this is another new term for you, conservation. Okay, conserved. Conserved. So which parts of the sequence are conserved means that it did not change. It was saved during the evolutionary history. Some part of these sequences may have remained the same, but other parts may have gone under some mutations and have changed. So the parts that did not change are called conserved regions. And probably there's an evolutionary reason behind this conservation. Because usually functionally important sites, functionally important regions of this DNA sequence, they need to be preserved. They need to be conserved during evolutionary history, because since this is functionally important, if there's a mutation in this region, that new mutated species will not survive. So for survival, if a certain region is preserved, is conserved in the evolutionary history among many different species, it probably means that this region has certain importance. It's for this gene to perform its function, this part is pretty important, so did not change if a mutation here will not help the species to survive. But some other parts, so not every part of a gene has equal importance. Some parts may, even if you mutate certain parts, the gene may still function. And biologists are still doing such kind of experiments in their labs. When they discover protein, for example, they do this is a very routine experiment, I guess. They mutate a certain amino acid to another amino acid and see whether this protein is still functioning. By doing so, they can identify important regions in this protein sequence, and they can identify maybe certain drug target sites. If you target a drug to that site, you disrupt the protein, for example. So it's very important. So a gene sequence or a protein sequence does not have equal importance on all sites. And after we find homology, the sequence alignment tool helps us to find these regions. Which regions are similar in these two sequences, which regions are different. So that's why we do sequence alignment. The process of sequence alignment is basically given a pairwise. The process of pairwise sequence alignment is given two sequences, protein sequences, and do not forget they may be of different length. One protein sequence may be 100 amino acids long, the other may be 300 amino acids long. So we are not trying to align the same length sequences here. We have two different strings with different lengths, and our goal is to find a certain substring, and we are also not looking for exact string matching. We are looking for subtle similarities maybe sometimes. So our goal is to find these alignment means that if a certain region is conserved, putting the conserved regions, the actual correct conserved regions on top of each other, and these similar parts, to align these similar parts, we may need to put gaps, because in order to align a 100 amino acid length protein sequence with a 300 amino acid length sequence, you need to, you cannot align a short one along with respect to a long one, so you need to put some maybe gaps in the short one, like as fillers positions, so that a substring may be aligned to this region, another substring may be aligned to the end of to the beginning of the protein sequence, for example. So we may need gaps in this alignment process. So I'm going to show you a lot of examples, and I'm going to describe the problem formulae as well. But to describe the problem formulae, we need to talk about scoring matrices. So I'm going to come to that. So let's go over quickly again why we need sequence alignment, because we have a lot of DNA sequences, we have a lot of genes. So when a new gene is discovered, our goal is to find out of these related species, for example, is there a similar gene to this newly discovered gene? And the databases are growing very rapidly. There are many sequencing projects that are in progress, as you have seen yesterday, and there are a lot of them which are completed or in assembly stage, but we have a lot of sequence data. And all this data is available, most of them publicly. You can go ahead and access these data sets and do searches, find similarities between species. And maybe using these similarities between the genes, we can drive the evolutionary relationships between organisms. This, for example, which organisms are we most close to in terms of these genetic similarities? Why is this important? Because if you're going to develop a drug, you don't want to test the drug directly on humans. If it's a new drug, so you need the clinical tests, you need to test it, so in animal trials, for example, you need to, your trials on animals, in order for your results to be generalized to humans, that animals should be as close as possible to humans, because the side effects are going to be most similar. You need to have this similarity, otherwise you cannot generalize your conclusions about this drug, whether it's going to be harmful to humans or not. So this is only one part of evolutionary relatedness, and sometimes considering the whole genome, maybe if we consider parts of the genome, we may find ourselves similar to some other very low-level organisms as well. So sometimes we do this evolutionary relationship based on a single gene. If we just look at, so this evolutionary relationship, this tree, this evolutionary phylogenetic tree, can be drawn using different criteria. By looking at the whole genomes, having a certain quantitative measure between similarities of whole genomes, or by looking at a specific gene, okay? For example, the globin. By looking at the globin gene, we can understand the similarities. Maybe if we just look at the globin gene, this tree may turn out to be a different tree. Okay, so, but understanding these similarities relationship, how similar these globins, for example, if you go to this tree back again, okay, for example, this shows that the mammal alpha globin is closer to snake alpha globin compared to the frog alpha globin, because this speciation event happened later in the evolutionary history. So this speciation event happened earlier than the speciation event 3, which means that at this branch, the mammal alpha globin and snake alpha globin shared some similarities. The differences between mammal alpha globin and snake alpha globin occur in these short branches. So there, mammal alpha globin is more closely related to snake alpha globin compared to frog alpha globin. Okay, so these kind of, the thing is, the bad thing is, we do not know exactly this evolutionary tree. Okay, this tree is not known to us. And this, the phylogenetic analysis, there's this area of bioinformatics which tries to construct these trees based on the information we have. So what is the information we have? We know the leaves, okay? We have these leaves, unordered, unsorted. I mean, we just have a sequence of, a set of globin genes. We have their sequences. At that point, we do not know if frog is close to us or snake is close to us, okay? They're all, somebody gives you these globin sequences in a bag. You don't know which one is related to which other better. And the goal of phylogenetic analysis is to draw such trees which would be most close to the real evolutionary tree. But that will be very difficult to measure how close to the, because again, as I said, sometimes we may not know the actual tree. So, and to derive such trees, sequence alignment is also very helpful. If we can find pairwise relationships between, in this bag of sequences, with the help of sequence alignment, we can compare each pair of sequences to each other. And we can say, out of these, for example, n square pair, if we have, how many here? 7 sequences. We are given 7 sequences here. Out of these 7 sequences, how many pairwise alignments are possible? 7 choose 2, which is 7 times 6 divided by 2, which is 26, right? So we have 26 pairwise alignments. There are 26 possible different pairs between these 7 sequences. So if you do 26 pairwise alignments, and it may turn out that if you sort the results of these pairwise alignments from the most similar pair to the most distant pair, probably the most similar pair are going to be neighbors in the branches of this tree. Okay, so you can devise an algorithm based on the result of the sequence alignments. You can devise an algorithm to draw this tree, and we are going to see it like maybe 3 weeks from now. We are going to see some algorithms, neighbor joining and UPGMA algorithms, which exactly does this. Okay, given these pairwise scores or pairwise distances, whatever you compute, it processes this pairwise similar to our distance matrix and comes up with a tree like this. Okay. And also I mentioned not every region of this sequence alignment, mostly if you identify certain homology with a gene, it turns out that most of the time it's not similar to a single gene. Okay? If you find homology, you find homology in many different species. So there may be a family of sequences that is related to you. And in that case, after you find this family of sequences, the next thing a biologist does is to multiply align them. It's called multiple alignment. It's like an extension of pairwise sequence alignment. Instead of a pair of sequences, if you know these 5, how many we have here? Like 8. Okay, we have 8 sequences here. It is mouse, chick, chimp, human, dog, rat, puffy, and zebra gene. But what gene I don't know. Okay, they are from these 8 different species. And if you find these sequences are related from these 8 species, you multiply align them. You put these 8 sequences on top of each other, revealing similar regions. And the more sequences you have that you think that are related, the better you are going to find the conserved regions. For example, there may be a certain region, or for example, I think these are gaps, or so this is a result of a certain multiple alignment tool. This is the output graphic. So each different multiple alignment tools provide different representations, but at the end, all they do in common is reveal conserved regions. Okay, this part is the same in all 8 species. There's a certain block. Sometimes there may be a block of amino acids that are similar. Sometimes a single amino acid may be important. Okay, the alignment of a single site, single region may be important. So aligning them on top of each other reveals these similarities between these different species. And the sequence conservation, conserved regions are related to the function of this protein. So you can also, by looking only at those conserved regions, by looking at whether there are certain known domains there. So, and this term domain is another new term for you. So whole protein sequences may be composed of substrings, subsequences, in which are responsible for a specific function. Like a statement in consider a C program, you have a function. This function is the whole protein sequence, but inside that function, there may be certain subparts. For example, the first of the function reads data from a file. The second part processes that file, for example, processes that data. So different parts of, within the same function in a C program, within the same function, there may be regions which are responsible of a single, more atomic function. We have similar things in proteins. There may be subsequences in a protein sequence which is responsible for an atomic chemical function. For example, binding sodium, for example, or binding potassium. There may be a whole protein. In total, it may be doing a more advanced thing, this protein. But there may be a certain pocket, certain region of this protein which is used to bind potassium. And we call this part, for example, potassium binding domain. So this subsequence is for that. This is just an example. This is called domain. And by revealing these conserved regions, we have domain databases, such domain databases, and querying these conserved regions in such domain databases, we can understand maybe that conserved part, conserved sequence, may be a known domain. So for example, if that conserved region resembles very much like a potassium binding domain, we may say that, okay, then this, one of the goals, one of the functions of this protein is to bind potassium. Okay, we can understand, after we find these conserved regions, we can do this. Otherwise, looking at all possible subsequences in the protein and querying in the domain database, it will be very difficult. I mean, querying meaningless things in domain databases will not make much sense. But if you find conserved regions, if you have a clue, if you have a strong clue about a region being important, then you can take this region and query in another database and see whether it has a specific function, whether it is part of a known, similar to a known domain. Therefore, it's very important sequence alignment to predict functions of proteins, to find homology and construction of these evolutionary relationships, construction of phylogenetic trees. And also, it's important for assembly of genomes. And remember, I said when we are trying to find whole genomes, what we do is we have fragments of these DNA, and our goal is to solve this puzzle by putting these fragments together. And the tool that makes this happen is to find these fragments need to have some overlaps so that we can combine them. Okay? Imagine, I mean, when you are doing a puzzle, you need some additional information to put two pieces together. Okay? In real puzzles, this information is that they have some complementary shape so that you can put these two pieces together. And in DNA sequences, the matching that is going to put two different DNA fragments together is their overlaps at their ends. Okay, at the end of one sequence, if there's a large overlap to the beginning of another sequence, you can put these two, you can join these two DNA fragments together to form a larger fragment. And if you continue doing this, you are going to get the whole assembly. I mean, it's not, there are a lot of other problems related with this. It's not that simple because you are going to have overlaps with many sequences, which one to choose to join. So there are many difficulties, but it's based on this simple idea. We need to have overlaps between small DNA fragments to join them to larger segments. So shotgun assembly also requires sequence alignment. And finding motifs, these conserved regions, these conserved smaller subsequences, they're also called sequence motifs. A certain sequence of amino acids which mean a specific function. Okay, for example, if you see a certain, it's like a regular expression. If you see this amino acid, it means this function. So this is called a motif. So identifying motifs with the help of multiple sequence alignment is important in this area. And now, how are we doing on time? Yeah, we still have 10 minutes before the break. So the sequence alignment can be described as a procedure of comparing two or more sequences by searching for a series of individual characters that are in the same order in the sequences. So this definition may be difficult to understand first. So, but what it tries to do is, some certain keywords. So if we are comparing two sequences, it's called pairwise sequence alignment. If we are comparing multiple sequences, more than two sequences, it's called multiple sequence alignment. And the main idea is to provide a matching between these two sequences. As you can see, this is an alignment, and this alignment tells us that this T, which is the third character in the first sequence, is matched with the T, which is the first character of the second sequence. And this A, for example, is matched to this. Why didn't we match this A to this one? Because this A was matched to this one. But maybe we could have, there are two A's here, okay? This A and this A, they could have been matched to this A as well. So as you can see, we have many alternatives in this matching. This matching is not very easy to find. But at the end, our goal is to, an alignment, a final alignment, is a matching, a one-to-one matching between the amino acids or the nucleotides, between these base pairs of the DNA sequences or amino acids of protein sequences. So our goal is to provide a matching, saying that this T, the second T, matches the second T in the second sequence. Okay, so this is alignment. So I say one-to-one, so and it has to follow certain rules. It has to be one-to-one because an amino acid, a letter here, a base pair on an amino acid, has to be matched either with another amino acid in the other sequence or has to be matched with a gap. So actually this is also here, this G is matched with a gap here in the other sequence. We put a gap against it. We cannot leave any sequence unmatched in global sequence alignment. So sequence alignment also has variations. In global sequence alignment, the goal is to match everything in every letter in both of the sequences. Nothing is left out. Here, if it's a global sequence alignment, this first G and first C are matched with gaps. We need to put gaps against them. And another important thing in this matching is that we need to preserve sequence order. Meaning that if you have matched this A with this A, it means that anything that comes in the first sequence, anything that comes after this A cannot be matched to a letter before this A. And we cannot have crossings like this. We need to preserve sequence order. If you decide to align this A with this A, it's like let me have pointer options, yeah. Okay, if you, for example, if we have decided something else, if we decided to match this A with this A, it's like I have divided the whole problem into two different parts because you are not, after you make this decision, okay, I lost the pointer, yeah, it's here, it's a very small dot. So if you decided to match these two A's together, it means that you cannot match this part with this part. You're not allowed to do it, okay, it's not allowed. If you match these two sequences, A and A with A, the second part of the first sequence cannot be aligned with the first part of the first sequence. So it's like after you make this decision, you're left with another sub-problem to align this with this. Okay, this is how we are getting to the dynamic programming solution now. So it's like we have divided, it's like a divide and conquer approach. If you're sure, I mean, if you make a decision, okay, this A is aligned with this A, then the problem becomes solving the alignment of these rest of these subsequences. Okay, where is? Yeah, it's here. Okay, so we are not allowed to, after this match, we are not allowed to do a match like this. It's not allowed. So we need to preserve sequence order. And everything needs to be matched with other things, with another amino acid or a gap. So we can either, we are going to match every letter with either another letter in the other sequence or with a gap. Gap is also sometimes denoted by a dash like this. Okay, I'm going to get back to this arrow. So these dashes, they denote gaps. And this is a good example which shows that you can have gaps in both of the sequences. It's not like that you're always going to have gaps in the shorter sequence. You can have gaps in the long sequence as well. So if you think about an alignment, if you have a sequence of length n and a sequence of length m, what is the maximum size, maximum length of the alignment? Okay, the length of the alignment here is, for example, what is it? 1, 2, 3. I'm not going to count it, but the length of the alignment is from here to here is the length of the alignment. So this is, if you count the characters from here to here, that's the length of the alignment. So if you have a sequence of n characters, if a DNA sequence of n base pairs and another DNA sequence with m base pairs, what is the maximum length of the alignment? Yeah, n plus m, right? Not minus 1.
Just one character, we match one character.
We don't need to match any character. It's not a requirement, right? We don't. I mean, it's not a requirement of the alignment. I mean, it may be so that. So this is the first sequence of length n matched against all gaps, and this is my second sequence of length m matched against all gaps. This is a valid alignment, okay? Although it's very nonsense alignment, okay? You're not going to have alignments like that. I mean, it doesn't. It says that they are not matched. Nothing is matched. They are totally different. Maybe to make that statement, okay, they are totally different, 0% similarity, you can have an alignment like this, and the alignment length is going to be m plus m. I mean, that's the theoretical maximum, but in practice, we do not have that long alignments. But that's the, so we are, and what is the minimum length of the alignment? Exactly. So it's the minimum length of the alignment is the maximum of m or n. Okay, whichever is maximum, that's the minimum length of the alignment we are going to get. This is in global alignment. Okay, so we are talking about global alignment in which every amino acid in one protein sequence or DNA sequence has to be matched against another amino acid or a gap. We have the other variation we have is local sequence alignment, in which we are not interested in the whole similarity, whole sequence similarity, but maybe there's a subsequence. If you look at the whole sequences, they may not be similar at all, but there may be a subsequence in both of these sequences which are very similar to each other, and the alignment concerning only those subsequences is called local sequence alignment. And we are going to see algorithms to find both global sequence alignments and local sequence alignments. But let's have a 10-minute break first, and we are going to continue after the break.